السلام عليكم. And this is the beginning of the second semester, and we will start with chapter number 10, which is the blood. So, what do you know about the blood? What's the blood?
Connective tissue.
It is connective tissues. Huh? What type of tissue is it? What is there? What is the component of the blood? What do you expect that you will study? Red blood cells. So the cells. So it contains some cells. From the cells, red blood cells. He mentioned. What other cells?
White blood cells.
White blood cells. Is there? Is there any other cells? Plasma. It is another component. So it is component from liquid component, and the cells, which is the white blood cells, red blood cells, and there are another one, which is the platelets. So the objective for this chapter, in this chapter, we will start with describing the component of the and the composition and the volume of the whole blood and its function, describe the composition of plasma and discuss the importance of it in our body. Then the third objective, describe the cell type making up four elements of the blood, the major function of each type of the cell, and describe the hemoptysis, which is the blood cell formation, how the blood cells are formed, describe the process of hemoptysis and the importance of blood clotting also, homeostasis, sorry, and describe the A, B, O, and RH grouping of the blood. Then explain the base for blood transfusion and blood transfusion reaction. So those are the components, those are the objectives that we will cover during this chapter. So the blood, it is for the transportation or the transport of everything that must be carried from one place to another within our body. So all the nutrients, all the blood gases, all the waste products are transported through the blood. So nutrient, waste, hormones, and also body heat, it is transmitted through the blood. So the blood is having the capability to transfer all of those components. The nutrient, after it is absorbed from the digestive system, it will be carried by the plasma and it will be sent to the cells in order to get its nutrient and to start the process of metabolism. The oxygen, it is taken from the respiratory system through the alveoli and taken combined with the red blood cells and carried out to each cell of our body. Hormones, it is also taken from the glands and sent to the target organs that it will act on it. Carbon dioxide, it will be carried out from the tissue after tissue metabolism as an end product, and it will be taken out of the body through the respiratory system. So the blood is having all of those functions in our body. So, and each cell it is the blood also it has not only the process of transport, also protection, immunity is there in our blood. So the physical characteristic of the blood, if we are coming to the physical characteristic, what we mean by the physical characteristic here? What are the physical characteristic of the blood? So it is liquid or solid? It is liquid. So it is a sticky, opaque fluid with a metallic taste. The color of that fluid, so it is a fluid, but it contains cells. It is the only tissues that is liquid in our body. The color of it is red. It is taking its color from what? Which portion of blood it will give its color?
Hemoglobin.
The hemoglobin or the red blood cells. So the color it is given by the red blood cells. And also it is affected by that color, it is affected by if the red blood cells is carrying oxygen or carrying carbon dioxide. So the oxygen rich blood is scarlet red, it is bright red in color, but the none or when it is carrying carbon dioxide, it will be of dull red. So the redness, it will be dull, or it will be to some extent dark. So this is the difference. So when you start to take blood from the patient, if you will see that color when it is taken from the venous blood, that the color is dull, dark. If the patient is having some respiratory problem also, you will feel that it is more dark because of the high amount of carbon dioxide in that blood cells. So it is heavier than water, 5 times more viscous also or sticky. So it has viscosity. So it is not that like water when regarding the viscosity. Water doesn't have that viscosity or sticky. It is not sticky, but the blood is sticky. We will come also to the pH of the blood. The pH of the blood, you took it in biochemistry. Did you remember what is there? I will cover it, but it will come out. Did you remember the pH of the blood?
Yes.
Yeah, how much is it?
7.35 to 7.45.
Yes, so it is 7.35 to 7.45. That specific pH, it will help for the action of all hormones, all enzymes in our body. If the blood become acidic or alkaline, in this case, there will be interference with those processes. So it is the main and the most important to have it regular in order to maintain our body working in a normal process. About the amount of blood in our body, it is from 5 to 6 liters in our body. So, as an adult, the amount of blood in an adult, it is from 5 to 6 liters. And it is the composition, it is about 8% from our blood. So this is the blood. The composition of blood. The blood, it is the only unique fluid tissues in our body. So no other fluid tissues is there. It is in the human being. It is thick and homogeneous also. So the combination of that blood, or the it is homogeneous. It means that all the component is equal in each ml of the blood. If we are taking it 1 ml by 1 ml, the same component will be there in those blood component that we are taking. So it is a homogeneous. Homogeneous, it means that it has an equality in the component for each single drop of blood. The blood component, it is a complex connective tissues. It contains living cells, which is the form element of the solid element of the part of the blood, and non-living matrix, which is the plasma. So it has two parts, the plasma and the cells. And collagen and elastic fibrin are absent but dissolved protein. They are dissolved protein in the plasma because and it will become visible as fibrin strains when we have injury and the bleeding came out. So in this case, clotting will be formed and fibrin will be activated. We will come to this issue and we will discuss it later in the process of the clotting. In the blood and its component, when we are taking blood, as we draw a whole blood in a tube, and I think all of you went to the lab for some kind of test, and they took blood from you. When they are taking blood from you, you see a different type of tube. The difference is in that you can observe easily, which is the cover of that tube. Either it will be pink or blue, or it might be red in color. There are different kind of color, and sometimes it is according to the company, but also it will indicate what is there inside the tube. So if we are taking a whole blood, usually for doing a CBC or taking a blood study to see the cells, and in this case we have to have an anticoagulant within the tube in order to prevent coagulation, because the blood, when we are taking it out of the body, it will be within minutes, it will be coagulated. It will be clotted. So in order to prevent that, they are having a tube which has anticoagulant. They will mix it, then they will put it in a centrifuge to separate the cells from the liquid, from the plasma. So you will see, if we are taking, and I have a specimen, but I forget it in the office, I will bring it and I will show it to you, that specimen. You will see that around 55% of the blood is plasma. So around 55 of the blood, it will be plasma, which is the fluid component of the blood. And then you will find a very thin, buffy coat here. You have to use your eye and focus in order to see it properly, because it is a very thin coat here, which is the white blood cells. Then we have the rest of it, which is around 45% of the whole blood. It is the erythrocyte, the red blood cells from the normal blood cell. So hematocrit or red blood cells, it is the composition of it, or within our blood, it is around 45%, and the 55% it is a plasma. So the plasma, the major constituent of the plasma, it is 90% water, which is, you know that water, it is a good solvent because of characteristic of water. And if you remember in biochemistry, the water is a polar component. It has a polarity. And because of that, it has the ability to carry the elements. So 90% of plasma volume, which is solvent, carrying other substances, and also absorb heat. And you know that also the water itself, it can absorb the heat and keep the heat within the body. Because of that, also the blood is transferring heat and distributing heat within our body. The other elements in the plasma also is the electrolyte. Electrolyte like sodium, potassium, calcium, magnesium, chloride, bicarbonate. Those components, you remember also the osmotic pressure, osmolarity, and the balance, and the buffering system. Which one is considered as one of the buffering system from those components? Which one in the plasma?
Potassium.
Sodium, potassium.
Bicarbonate. Bicarbonate, Malcolm. Bicarbonate, what is it? It is one of the components that is an anti-acidic, or it is a weak acid, a weak base, sorry. Weak acid, base. So it is related to osmolarity, pH, and buffering, regulating the membranous permeability. Because of that, we took biochemistry in order to understand what will be going on in our body. Then we have plasma protein. The plasma protein, albumin, fibrinogen, the globulin, and each one of them is having a function. They are also, from their function, the osmotic balance, also the pH buffering, also from the albumin, clotting, which is the fibrinogen, it will act in the clotting, defense mechanism and formation of antibody, which is the globulin. It will deal with the formation of antibodies, and also liquid transportation. Substance transport by the blood. As we mentioned in the beginning of the lecture, all the substances are transported by the blood. Nutrient from glucose, fatty acid, amino acid, vitamin, waste product of the metabolism, like urea, uric acid, all of them, they are transported by the body. Respiratory gases, the oxygen and carbon dioxide, hormones, steroid and thyroid hormones are carried also by the blood. So all of the transport within the body are carried out by the respirators. Just sit here. So this is the liquid part that we measure. The second part, which is the cells. So the cells, or the form elements of the cells, and we said that it is around 45% of the whole blood. We said that mostly it is consists of the erythrocyte, which is the red blood cells. It is around 4 to 6 million per cubic millimeter. So the main function of it is transportation of the oxygen and help in the transportation of carbon dioxide. Carbon dioxide mainly it's transported by what? By the blood, by the red blood cells, or by the plasma?
Red blood.
Plasma. So mainly transported by the plasma. Yes, some of it is transported by the
RBC.
red blood cells. You have to refer back to the biochemistry. The leukocyte, it is around 4.8 to 10.8 thousand, the white blood cells. It related to defense and immunity. So the main function of it, it is the defense mechanism in our body and immunity. There are many types of white blood cells. We have the basophil, neutrophil, eosinophil, and leukocyte and monocyte. So those are the blood cells, the white blood cells. We will also come to them, and we will divide them to granulocyte and agranulocyte. We will also see the function of all of them. Then we have the platelets, which aid in the process of clotting. So, which is 250,000 to 400,000 per 1 ml. So, this is the blood, the cells, and the amount and their function. So the main function of the red blood cells is transport of oxygen and some carbon dioxide. The main function of leukocyte is for the process of immunity. It is, we will come also to the type of immunity, the cellular immunity and humoral immunity, and the antibodies, and also phagocytosis. We will come to that. Then we have the platelets, and the main function of the platelets, it's related to the blood clotting. So now you understand the component of the blood?
Yes.
So it's composed from two main parts. What are they?
Erythrocytes, leukocytes.
Plasma and blood cells. The plasma, the function of plasma is mainly for what?
Nutrition.
For one of them is nutrition, so it is mainly for transport of and dissolving of all components within the body and transferring of those components either from the cell or to the cell. So the main function is transport of the component from to the cell or from the cell. So those are the main function of this part. Now we'll come to description of the cell type making up the formed elements and the major function of each, and describe the hematopoiesis or the blood formation. The formed element of the blood, as we said, we have the erythrocyte or the red blood cells, the leukocyte, and the platelets. So, erythrocyte, red blood cells, white blood cells or leukocyte, and the platelet cells fragment. The red blood cells, if we come to the shape of it, it is like a cone shape, and it is concave from each part, each side. The platelets or the thrombocyte, it is very small elements of the cells, and we said that mainly it is for aiding in the clotting. So the erythrocyte or red blood cells, and white blood cells, and we said that there are two types of red blood cells. We have the granular leukocyte and non-granular leukocyte, granulocyte or agranulocyte. The granulocyte or the granular, see the difference in the picture. What do you observe with the difference between those three and those two? What's the difference that you observe? It is clear. See here, what's the
Point.
and see. Is it present? Something is not present here.
No present.
Sorry?
No present point. Only liquid.
So, in the cytoplasm, there is what we call it, we call it the granulocyte. The granules. Granules is there. Here, there is no granules. So, it's granular and non-granular. Granulocyte and agranulocyte. So, this is the difference between the white blood cells. The granular leukocyte, it is the basophil, neutrophil, and eosinophil. So those three types of cell, it is granular cell. The non-granular leukocyte, it is the lymphocyte and monocyte. So monocyte and lymphocyte are not having those granules. So when you are seeing taking a blood film and try to see it under the microscope, you will see it like this. So you can differentiate between the white type of white blood cells, the granulocyte and agranulocyte. When you are going to do the CBC or complete blood count, you will find that they are giving you a number of or a percentage of how much you have from basophil, neutrophil, and eosinophil, leukocyte, and monocyte. And all of those numbers, if it is coming up or coming down, it has a meaning. So we can reach to diagnosis of the disease from the changes in the concentration or in the amount of the white blood cells. Whenever if we are having any infection, what's the first thing that we will look for, and it will be raised? It is the white blood cells as a whole. The white blood cells. Then, when we look at the percentage of each one of them, you can also, from that, you can detect what type of infection are you having. Do you have a viral infection? Do you have bacterial infection? Also, in some cases, even in case of cancer, there are some changes in the white blood cells. We will come to that. So, did you understand the importance of understanding how the percentage of white blood cells in our body and the amount and the increase of white blood cells? So it is important in order to give us a diagnostic measure for many diseases. Why? Because they are fighting the disease. Because of that, there will be a difference in their percentage when we are having any disease. But when our body is stable and in a homeostasis, in this case, it will be within the specific range. So the foreign element of the blood cells, as we said, we have the monocyte, we have those lymphocytes, neutrophils, the platelets will be a small elements, and mostly when we look to the blood cells, it's mostly the red blood cells. So this is the formed element of the cell of the blood when we are seeing it under the microscope. When it is seen under the microscope, so you can see the monocyte, the lymphocyte, and the neutrophil, the platelets, and the red blood cells. The erythrocyte or the red blood cells, it is, as we said, it is a small flexible cells, biocaved flat disc. Biocaved means that it is having concave from both sides. Function to ferry or carrying oxygen to the body cell. So it is taking the oxygen to the body cell. It is unnucleated cells. So it is the cell that doesn't have a nucleus. So it cannot multiply after it is released from the bone marrow. It will not multiply in the blood while it is there in the blood because no nucleus is there. So it is formed from the mother cell in the bone marrow. But after it is released, it cannot be divided. It doesn't have also mitochondria. Because of that, it could not consume oxygen from the oxygen that it's carrying. So it contains a very few organelles. Nature of red blood cells are sacs of hemoglobin. So it's like a sac containing only mostly the hemoglobin. It lack, as we said, there is no mitochondria, lack of mitochondria, do not use up any oxygen that they are transport. Because if it has mitochondria, it means when the oxygen is linked to it, it will be consumption by the red blood cell before it reach the other cells. So the cells, it will be deprived from oxygen. But because it doesn't have mitochondria, so the oxygen that it's carrying with it, it will reach home, it will reach the blood cells, the body cells, without touching it, without consuming anything from it. 5 million cells per cubic millimeter. So for each cubic millimeter, it has 5 million cells of red blood cells. red blood cells also contributor or major factor of blood viscosity. It is the one which is give the viscosity of the blood. هي اللي بتعطي اللزوجة. So sometime or in some person we might find that they are having severe anemia, especially if it is chronic anemia, kind of type of anemia. In this case, when you are taking the blood sample from him, you will see that it is a liquid that it is not that much sticky. You can say from just observing visualization, from seeing that specimen, that this person is having anemia. The blood is not thick in this case, not viscous, because the amount of cell in that blood is not. So this is the side view of it. It is around 2 microns, and if we are taking from both sides, it is around 7.5 microns. So it will be taking the hemoglobin. It is, as we said, it is a sac of hemoglobin, and it's used to link with the oxygen in the alveoli when we are taking the respiration. So the blood which is there around the alveolar sac, it will take the oxygen and it will carry it to the blood cells until it reaches the blood cell. Until it reaches the body cells. Then the hemoglobin, due to difference in the concentration, it will be released to the blood cells. We will come to that. We will explain it fully. This is this part when we are taking the respiratory system, in both in fundamental of nursing and in anatomy and physiology. So we will come again to this portion, how the oxygen is taken and how it is linked with the red blood cells, and after that how it can be released from the the red blood cells to the body. So the next session I will give you 10 minutes break, and the next session we will continue, inshallah.